This is an overview of DNA replication. And you'll see on the inset that it's not just that there's one leading strand and one lagging strand. Depending on which fork you're looking at, one strand is the leading strand and one strand is the lagging strand. So each of the strands can be leading or lagging depending on where it's located. On the right side, you'll see leading strand with continuous synthesis is on top and lagging strand with discontinuous synthesis on the bottom. However, on the left fork, the lagging strand's on top and the leading strand is on bottom. We see these two different directions because replication is bidirectional. Of course, there are always exceptions to things that we speak of as fixed, but almost always there will be a replication bubble with synthesis going in both directions. We'll focus on the view of this picture on the right-hand side of the overall, with the leading strand at the top and the lagging strand at the bottom. So we start with helicase, which opens DNA. It separates the two strands from each other. Single-strand binding proteins come in wherever there are single strands on either the leading strand or the lagging strand. DNA polymerase III with rapid, continuous synthesis on the leading strand, but on the lagging strands, primase starts primers. DNA polymerase can extend if there are large gaps, but often what you see on the lagging strand is DNA polymerase I, which was particularly good at removing the RNA primer, shown in turquoise. After that, ligase seals the gaps between the fragments. So we end up as products of this process with two identical DNA sequences, and each one of them contains a new daughter strand and an old parent strand. So each of these products is a hybrid with one new strand and one old strand. The sequences are conserved in both of these strands with rare errors.